Well, this is the point. So let's go there. This is electrobiology in a nutshell. That essentially the soil medium, although the surface of the earth is broadly positively charged, it has a positive potential. The actual soil mediums themselves, the particles, are broadly speaking neutrally charged, and this is why soil pH is always around 7.2. Basically, what we know as pH, so acidity or alkalinity, is either an excess of electrons or a deficit of electrons. Okay, and pH neutral is no electrical activity at all, so neutrally electrical. Now, the other thing about soil, especially the better soils, is that they're largely made up either of quartz or silica minerals, and that these quartz and silica minerals present A, an enormous surface area, but B, also an incredibly electrocapacitant, neutrally electrical surface area, which will catch hold of positively ionized water molecules, i.e. slightly acidic water molecules, carrying a slightly positive charge, and these will be bound to these soil particles. They'll be electrically bound to them, but they'll only be bound to them very weakly because they're not being bound to a negative charge, so they're not being stuck like two magnets, north and south of a magnet together. They're being kind of bound more like, what are they bloody called? Those post-it notes, yeah? So it's not like an Evo stick, it's like a post-it note. It's stuck, but it's not stuck very firmly. A slight pull will get rid of it.
Yeah.
Okay? Now, essentially, the root on the plant is like the sheath on the wires in your mains. It's an insulator. It's an electrical insulator. And we can consider the fluid inside the vesicles within the root itself as carrying quite a strong negative charge. They'll be quite alkaline. Now, at the surface of the roots, we have the pores. Now, because the pores of the roots are on the outside of a sort of an elastic pipe, that when that pipe shrinks, the pore opens. When the pipe expands, the pore closes. You see? So the pores in the root surface are literally self-regulating according to the level of hydration of the plant. So if the plant begins to dehydrate, the pores naturally open, and if the plant's well hydrated, then the pores will naturally close as the pipe swells and closes up the aperture. Now, because the pipe is effectively an insulator and the electrical activity is inside, when the pore opens, what it does is it presents a little localized hotspot of highly negative electrical potential. And these weakly attached, positively charged water molecules within the soil medium will be inexorably pulled towards it. And you can imagine the pore being like the end of a bar magnet under a piece of paper that you sprinkled iron filings on. Yeah? Where the lines of force will be greatest, nearest to the pore, but they will extend out from into the medium.
So the plant's roots could be drawing water from quite a distance away.
From an infinite distance, because it's subject to the inverse square law, that it will be drawing water from as far as it can influence on an exponential decreasing curve.
Exactly the same as you were describing before the break, to do with our atoms and charge. Yeah.
Precisely. This is how charge acts. So charge separation acts through the inverse square law, where for every 1 unit of distance gained, you have a 4 times increase in area of action and a 4 times decrease in the power of action.
You see, that's absolutely brilliant, because I often wondered how, when plants get thirsty, which, when they're growing as youngsters, they put on
like
some
a dry tree seeking water,
incredible amount of mass. And I was figuring that if the roots were only getting water from the thing they were in contact with, they were going to end up with dry roots pretty quickly, and that's a really good way of killing a plant.
And this is essentially where the hyphae, where the mycorrhizamal hyphae come into this. So the symbiotic fungi. I mean, when you look at a root system, it's actually only the thick, fibrous white roots in the middle that are actually the plant. all that fuzzy stuff around the outside that most people think are plant roots are actually mycorrhizamal fungi that have basically wiggled their heads in between the plates of the roots system and embedded themselves in one of the root cells and is feeding sugars from the plant, which the plant quite happily gives it. In fact, the plant will send sugars down once it detects the presence of a fungi, of a fungal head. Now, how does the plant know to send sugars south and ionic water north? Well, it doesn't. It's because the charge that the head of that hyphae carries is strong enough that it will draw the negatively charged sugars towards that hyphal head. And in return, the hyphae is connecting the plant to something like 1,000,000 times more surface area than the roots could ever contact in 1,000,000 years. And moreover, the hyphae grow at such an incredible rate. I mean, literally, I've been watching stop frame animations, and mycorrhizamal hyphae look like bloody lightning storms in these things. They are that fast. Admittedly, they're like hour stop frames. So, I mean, it's 1 hour from one image to the next. But I'm seeing things occurring within those stop frame animations that just look completely unbiological. The speed that they happen at, it looks more animal than...
And where do they come from, these mycorrhizal fungi, in a pot? You know, you've got a pot.
Well, essentially they're
specific to
everywhere.
plants as well, aren't they?
The spores of mycorrhizamal fungi are in every single mass of dust that you've ever seen in your life. Will have been billions of them. Certainly, if you go to like a woodlands, all of the dirt, all of the dust that you see blowing around by the sides, a vast majority of that will be fungal spores. And every time that you see a wood pile with a big fungus poking out, a big mushroom poking out the top of it, well, that entire wood pile is in fact the mycelium, the fungal hyphae that are the actual mushroom. And what you're seeing there, what we call a mushroom, is in fact only the fruiting head of that mycelial mass. And the mycelial mass will literally be occupying every inch, literally every square inch of that wood pile.
And it will only it will express itself in the soil with or without the presence of are these...
Well, no, they actually respond to their symbiotic partners.
Okay, right. They need the food.
So the specific fungal hyphae will seek out their selective symbiotes. So specific species like particular symbiotic relationships with specific mycorrhizamal fungi, and somehow the two manage to find each other.
And how many of these mycorrhizal fungi are there? I mean, are we talking about a dozen, or are they specific to individual plants?
There's about 5 kilometres in a cubic centimetre of soil.
Mm-hmm. But are they specific? I mean, I gathered that, like the example of a wood, I'm told that trees of a given sort within a wood will all be connected by a specific sort of mycorrhizal fungi, which is doing exactly what you're describing.
The forest floor in those woods, as you correctly say, each species will have its own hyphal mat, and the entire forest floor will be made up of that mat. And that those trees are not only in communication with all of the soil and all of the water in the soil, but it now appears that they communicate quite literally electrically through that network. And if there's a dying fellow, if there's an ailing fellow amongst their group, the trees are known to donate their sugars to the one that's ailing. Now, again, I would suggest that that's an electrical property.
Blimey.
That the one that's dying is having problems with its electrical circuit, so the other plants suddenly find on their network a negative draw, where before there was an excess of positivity, so their food is inexorably drawn towards their ailing cousin.
So a wood will contain lots of different sorts of this fungi to cover all
and
the
all
plants.
growing and dying at an incredible rate as well. But the normal hyphae only lasts about 4 days, and then it begins to go green and die back, and a new one will replace it. So this is another thing as well. These are exploratory creatures. They literally explore every inch of the soil medium and overtake it. And because they've got a growing tip that connects with these positively charged ionized waters, that effectively makes each one of those hyphae a one-way electrical circuit, always pointing towards the plant, which is another thing that I mentioned to you off air, wasn't it? Was that in the chat, was that every time I measure a voltage in my system, that I can take that reading from the plant to anywhere in the soil, and it will always be the same direction voltage. It will always be from the soil towards the plant. according to the rules of my electrobiology, according to the canopy being positively charged and the the roots being negatively charged.
Brilliant. Brilliant.
So effectively, what we're seeing is we're seeing a sort of a protozoal and amoebal soup within the soil medium. What we think of as soil isn't actually soil. It's small particles of soil that are surrounded by protozoa, which are all feeding on it. Those protozoa exude a kind of jelly snot that is the binding agent that actually sticks soil together, that turns soil from dust into soil. So every single piece of soil that you've ever seen in your life wasn't actually soil. It was a tiny little particle of dust surrounded by 100 or 1,000 protozoa in various stages of either life or death. Now, the protozoa, I would suggest, in their billions, provide a tiny electrical charge, which will also have its mechanism in regulating the water pH of any water that comes into contact with them. Okay, so if they make their environment slightly acidic, then any water that passes through that environment will pick up that ionization and will become acidic too. So essentially, these are like little electrolyte generators and generate the electrolyte for the soil. The hyphae come along looking for water and plug into that now positively ionized electrolyte. And because they've got one end in a positive circuit, that means that the other end has got to be negative. It's got no choice. So the positive ions will inexorably move their way along the strand of the hyphae, up the pipe of the hyphae, and be delivered into the core of the root, right where it's needed, into the root cell itself, where a charge of positively ionized water will now be delivered into a water circuit that is connected to the positively charged canopy, but is in a negative charged state. So the positively ionized water is literally given an electric shunt up to the canopy. And it's not until it breaches the soil that it's then taken over by sort of flow and evaporation and things like that begin to kick in and have a minor effect. But the major effect in sap transport and in water acquisition is an electrostatic effect.
God, it's incredible, isn't it? It's
okay, and so we'll kind of get past this, and so we've done the root system now. Above the ground, things start to change slightly. Above the ground, we're now looking at first of all, the shapes of the roots. The tap root will always go down almost perpendicular, and the reason for this is it's given like a little charge of sort of lodestones, or like little heavy bob weights in the tip, and this effectively acts like a pendulum pointing the way down. And so roots will always grow inexorably, broadly speaking, straight down, or at least the taproot will. Now, the reason why the side roots branch off at the angles that they do is, again, charge separation. That because the growing tips of the roots carry a negative charge exactly the same as the growing taproot, those charges repel. And they repel at very specific angles. So the actual shape, the actual pattern that we see the roots taking as they migrate through the soil is dictated by charge separation, and they will always maintain a correct charge separation with their negatively charged neighbour, simply according to the rules of Faraday's and Maxwell's and Gauss.
right, so it's doing both jobs at once. It's attracting what it needs, and it's maintaining space for itself through the same.
Yeah. And now, so we get above the soil. And now, effectively, what we have, let's take it from germination of the seed. So on germination of the seed, the growing tip doesn't do a damn thing. It just sits there in the seed, all folded up. The root starts a bid for downwards because it's got the lodestone and it knows which way is down. I suggest that the initial wetting of the seed is like a transistor switch event where on a transistor you switch the major load with a small signal wire, where you simply place a little bit of extra current into the silicon wafer, and that allows it to transmit the large current across the two big terminals. And I suspect that this is the sort of thing that's happening in biology, where we're seeing these large switching events that appear to take place all of a sudden, all at once, and from no apparent source, like seed germination. So I suspect that what's happening with seed germination, and certainly this lima bean